thank you very much. Nice to see so many of you. We don't need to talk about the importance of EMBnet. It has been just perfectly demonstrated in the last talk that I could partly be here. So sorry for being late. I had another meeting that I committed to earlier on. Anton asked me, can you just predict the future of bioinformatics or the field a bit? So as in Dutch we say that is like koffiedik kijken, which translates in English maybe to looking at coffee ground or something, meaning, you know, it's a big gambling business. We'll see what we get. But I'll talk a bit about history. That's easy at my age. Far more of it. And so then see whether we can connect that to some threads into the future. I have to apologize to some of the students who had a recent lecture with me in one of our master courses, Fundamentals of Bioinformatics. Some of the slides you'll see back here because I thought, hey, this is a nice base for this talk. So let's see if we can dive in it. Yeah, sorry, go back, it seems to work. Yeah, all right, so first point, lots of sciences, life sciences, maybe all sciences, have mainly turned into data sciences. So data is the all-important thing. And maybe also are becoming ever more interdisciplinary, bigger teams, articles where the first page is just a list of authors, or you get abbreviations of this consortium and that consortium and so on. So it's getting more and more. You know, more than 100 authors is not a really exceptionally anymore, right? So how do you do all of this? But another point is, which is probably also important, is that modern sciences are based upon data and data creation, data acquisition. And here is just a list. I'm not going to read it out because this will take an hour. You know, what is all there and what fields and so on. And, you know, so it's becoming more and more. So one of the things, a word that will be key, I think, is spatial size. Surroundings, because that gets even more important in bioinformatics in the old days. So we're scaling up. In general, we're scaling up to bigger questions, but also, you know, space was also was always a difficult thing. It will be there, but if you want to have an organismal look and so on, you need to take those dimensions into considerations. And there is a lot of development in that field, as you can already see here with some of the lists here. So even a lot of new stuff is coming up, you know, some combinations. You know, I found one joint RNA, protein, chromatin, spatial assays. What more do you like to combine stuff? So, you know, it will be, you know, we're all in good times, you know, lots of new things are coming up quickly and they're all very, very exciting. And of course, you know, to underpin why computer science and so on is always important, all of this stuff would not happen without computation. Okay, so as a boy, I was doing fishing and so on. And, you know, I did is using this, you know, with a float, right? And you stared for an hour at least. And maybe there was a fish, maybe not. So if you think about the field, how it has merged now, this was the old way. We were looking at specific problems, thinking about it in a specific way, developing specific knowledge. And now, say, with all of the AI developments, it's more like a big trawler and a big net, you know, ruining, of course, the oceans, getting all the fish on board the trawler, and then, you know, people go like, okay, this one, no, not this one, not this one, not this one, maybe this one, right? So it's not specifically going for a thing, but just, you know, have an overview. And, you know, there are very good aspects, but this is basically a trend. So we're thinking sometimes, right? And so, but anyway, I'll start, you know, stop being cantankerous at this point. Right. So, but to underpin the importance of AI, you can see here, you know, last year, there was a Nobel Prize for AI, and, you know, you see how important it is in our field. It was for a tool in our field like AlphaFold, right? Predicting protein three-dimensional structures, basically. And if you think about it, I've looked it up, you know, this is there is one has been faster, but this is the second short turnaround time. It's just 3 years, which is amazing, of course, for a Nobel Prize. I think in 1952, there was a physicist who had a parity violation, and that was a prize within a year. And there has been a Peace Prize for the Red Cross. You know, they were founded, and then immediately there was the prize, you know, to encourage them, I guess. So, but then this is a third overall, so not bad in science, I would say. And it stresses the importance that is given by a lot of people to AI technology. Oh yeah, your turnaround time. Anton, I have roughly half an hour. When do we want to go to drinks? Ooh, okay. At least I'll talk. All right, we'll see. Skip a few slides, I guess. All right, the Human Genome Project, and very quickly, you know, all of this, of course, was the first collaborative interdisciplinary life science project because the internet was there. You know, in 1995, you know, the owner of Microsoft predicted that the internet would be gone in 5 years' time, right? Well, how wrong can you be? But here collaborative might be taken with a bit of salt because it has been a big competition, a big battle between a very special character, Dr. Craig Venter, who was up against, you know, the allies, you might say, the Human Genome Consortium. And so they were in a race, and that race led to Bill Clinton, who was then the president of the United States, phoning up a few high-ranked scientists, asking them, could you use maybe 5 million to your lab to speed up, because I hear there are problems. The idea was, or the threat was, and this is the press conference, so all is well that ends well. You can see that here. But the threat was that Craig Venter, he was a bit frustrated about lack of funding, and he decided to start a company, Cellular Genomics. People were laughing at him. He will, of course, lose in the race and so on. And you know, he won't do anything, but he had the largest compute cluster before anybody knew it. There was a lot of venture capital, so he became a large force and a threat, because he said, if I will be first in doing the genome, you start paying me the money to look into it, patenting and so on. So that was, and there was no legislation at the time to say, this is ridiculous. How can I, you know, own your genome when it's sequenced, right? But there was nothing. So Bill Clinton decided it's maybe good to start a race and make that the consortium, the Human Genome Consortium, will be finished earlier than Craig Venter. So that's sort of, well, did it happen? Well, in the end, it did by 3 days. But more about this. So these are the two parties. The press conference with Bill Clinton, I just showed you the picture, was on the 26th of June in 2000. These two articles appeared in the same week a year later in Nature and Science, you know, just saying something about the competition still going on. And this was all highly orchestrated and so on. So a lot of talks. So anyway, Jim Kent, you know, an innocent bioinformatician sitting in his loft, he likes to program and not doing anything else. And, you know, people were saying, will he ever have a career? And then this happened. So so people were really in agony because the gap between the progress with Celera was so vast a year before the end that everybody thought it's all lost. Start paying Venter. And then, you know, this guy used a bit makeshift computer cluster and started programming like mad. And he built a project that was a product and a computer program that sort of worked and could create the first assembly. And you see a few sort of nearly funny dates. So Kent's first assembly, based on his makeshift program, was realized on June 22nd. And Venter, he thought, I can lean back because the gap is so large. But then once this came up, and he started you know, acting. So 3 days later, Venter published his genome. And then, you know, they said, now we're all friends. So I don't know how many telephone calls have been made at that time. But the next day, there was this press conference world over with Clinton and Tony Blair from England by a satellite connection. Okay, so this was seen, but he's still, I think, mainly sitting in his loft and doing programming. So some people just go on doing good things, right? Are not influenced by fame or anything like that. So, which is nice. So since then, you know, you see a lot of developments, of course, in sequencing technology. This is a bit older slide, but it still shows you. So the Sanger technique on the bottom there, that was used by in the Human Genome Project. Reads of roughly 1,000 nucleotides long. Well, after 900, you get some errors. And then you see the developments nowadays. Of course, it's all high-seq and so on still. That is maybe predominant in hospitals. You see the speed, it's logarithmic, so this is thousands of times faster, but the reads are short at 100 to 150. But the current exciting developments, of course, are with Oxford Nanopore and PacBio and so on, that have reached in the 10,000s. And the thoughts are now that with Nanopore, maybe we can start sequencing a complete chromosome by optical techniques. So there will be nothing lost anymore, everything there. So of course the error rate is still a bit high, but we won't need to wait very long before these things will be realized. So exciting, I think. All right. Oh yeah, I said that. Okay, so here's just to show you what it is. This is roughly a big American fridge, maybe, if you stand next to it. It's a bit higher than you, not by everybody here, but okay. And this is just a micro stick. This is this size, right? It's as big. And so yeah, you can go to any country to sequence. You will have a suitcase of other stuff with you. Okay, and you do all of the sequencing of local viruses or anything like that. Completely exciting. And the technology is even further advanced because this is already happening in 2015. Okay, so let's stop here for a moment. Do you really want me to go on? Because maybe it's fair to say that bioinformatics is a bit of a nervous field, right? There is all the time new stuff. There's never a boring moment, that's true. But every week there is something. 10 minutes. Let's see, then I'll skip half my slides, but okay. Anyway, so this is exciting, but if you want to get out of the field and do something at a slower pace, maybe Greek philosophy, old ancient Greek. Now, of course, very important, foundational, but knowledge comes up every now and then, so on, and the book is being written about it. So, you can leave and go there. Be happy if you want a slower pace life. Okay, I'm doing this one, and then I'm going to skip maybe a few things. So why is it nervous? So 30 years ago, for example, and that is also, we're in for surprises. 30 or 3 decades ago, a bit more now, by now, we already did sequencing and we found that there were all kinds of invariable regions in the genome that we couldn't attach any function to. There was no protein coding gene there and we didn't know, but it was totally conserved. And we know the evolutionary rule, when it's conserved, it's probably important. So something must, and I often say, if you find something you want to make yourself famous, give it a name, a good name. And if you don't understand something, give it a cool name. So hotspots, of course, is a great thing here. But then, a few decades later, microRNAs were identified, and many of those regions just coincide with microRNAs. And in that time, that's also maybe interesting, microRNAs have not been recognized. So the turnaround time for microRNAs is a lot larger. Finally, there was a Nobel Prize last year for it, but RNA interference technology was much earlier, although that's less important biologically because it was just more worked out than it was technology. And the Nobel Prizes are often given out for worked out sort of technologies, right? So, so this is maybe just interesting facts. But, the score is set straight with the Nobel Prize for microRNAs as well. Right, so welcome to the Jota world then. There's a lot of data around, so what do we do with it? That's I think part of the few more minutes that I may have. So I'm going to do lightning speed. This is showing that data is growing exponentially, right? That still is largely true about most sections of data, literature, protein data banks, genomics databases in general. Right, and then another point, you think it's growing very rapidly, but data also evaporates. Yeah, slide by Barend. Thanks, Barend. But you see here, this is Nature, I think, that the raw data, for articles, quarter of the data is lost after just 5 years. Data's, going astray, it's evaporating. And yeah, so, and then often, what's the reason? Oops, that was the link of my PhD student. We don't know where the guy is now, and the laptop is gone, right? So this is, again, the growth, exponential growth, that is true. And so growth is phenomenal. And here you have this old Moore's law for computation, where 1.5 years you have a doubling of speed and storage capacity. And then, for DNA, the doubling time, I don't know what it is now, but every 5-6 months, we is back the estimate, it doubles. So what do? This grow 1.5 years, they double, so we have too much data, right? So there is a fundamental problem here. And so how to solve that problem? maybe so compression of data might be something, or delete primary data, and that is often resorted to. Then we have to throw data away, nothing else we can do. And that has led in, as a result, first genomics, and then the primary data for genomics is terabytes easily. Of course, the genome is maybe 2 gig. DNA sequence is not a lot of data. But if you want to do it again or look back at what did we make an error, if you can't store the raw data, you need to store the DNA. So that's happening, and biobanking has come up largely 5 minutes. Okay. So very quickly, who can guess the molecule that is very efficient in storing information? You might know it. It's 4.2 billion. Oh, all right. So, okay. I won't do it. So anyway, so this is amazing. It's a great molecule. We all knew this. So reading out the information we're pretty good at. That's called genomics and so on. But storing information, writing DNA is a bit more problematical. Those stuff is coming up. George Church had his book about this PDF written in DNA. So that was a nice try. And so there are some early day attempts to do that, but let's see what the future will bring there. Right, yeah, all important, this. So another point is, we, of course, there are all these problems like how do we deal with data? Okay, and data is important. That's a very nice sentence by a monk, Ernest Dimnet. That even in the absence of data, you can be a genius, but you still don't get very far. And I think we can all agree with that. Yeah, then there is this point I wanted to make about respecting data. For bioinformaticians, you are dry scientists that respect the data. And there is one phenomenal guy who can attest to this fact is Amos Bairoch. He was there at the ISMB ECCB meeting this year, and he complained a bit, and I make a photograph of his slide. So he said, funding is going down for databases. How can that be if we just concluded that data is all important and everything is data science? And so here are, and he said, I asked ChatGPT what the reason is, and they came up with that. This is a nice one, of course, and this is really happening. So, yeah, so anyway, and then, of course, he's big on biocuration. People now think, and here you see again, this is very important to guard the quality of data. You see here, come on, so you're a failed scientist and you're lucky and it gets worse over time, right? So I thought I should show you this. Okay, that's Byron. It's not me. So, and then we have the other thing, fake news and all things happening today. And also, it's democratic to have data, right? In the old days, this was richer. Dutch families could buy this big encyclopedia. It would cost 2,500 guilders, which was phenomenal in those days. You could buy nearly a car in those days for that money. So it's amazing. And nowadays, so it's access to data for everybody, but how to use it, how to treat it, what happens. I'll look it up. Well, early 80s. Amos, you're, very good. He's there. Beautiful. Thank you. Yeah. I know a few more Amoses, but yeah, it could still be. Okay. Yeah, okay, so very quickly, this is an early champion on this. It's a gruesome picture because that thing he shows as a Christmas present to his wider family was the brain tumor that was excised from his brain during a 10-hour open wake surgery. So he had a story to tell, and he decided, oh, something bad happened to me. Where is the data? I would like to compare it. Can I have my data, please? And they said, yeah, we would love to give it to you because we like you. But there is no way we can do this, the regulations prohibit and, people tell us not to do it. So I'll skip a few slides, I promised. So he started shouting out, and he did this in TED Talks, and he was with President Obama, and he was in the New York Times. So this guy was a shaker, and he started saying, why is this not happening? Why can't there not just be a hospital button like, here is your data, and this is how to, so we're not in that world yet. Will we get there? And, we hope we will. And this is the man who unfortunately died because of that later on, of that cancer in his brain. I'm going to skip this. Vocabularies, ontologies, I'm sorry. This is for students, but okay, so linked open data. This is connecting. There is technology now to connect databases, and this picture shows you every every ball here is a database, and the orange bit is the life sciences databases. Now it's all much larger than the time this slide was made. So there is a lot of developments there, and this can lead to simple reasoning. Suppose the question is, can a bug cause malaria? And we know the answer, I think. So what you can do is then ask all these databases together, what do you know? What terms are there for the bug? What terms are there for malaria? And then you just look at the overlap. And that has led already in the early days to a lot of new insights and treatments for patients. Not malaria so much because that was too well known. So I wanted to talk a bit about FAIR data, which I'm going to skip. I can only tell you it started just 2016. That's not even 10 years ago. And now everybody talks about it. You could say that's a success. The management of that or the marketing has been quite good. It's by no means totally new, but the FAIR, findable, accessible, interoperable, reusable is a nice acronym for data. So that is this here. This was the paper in 2016, which is attracting a fair number of citations. So fair data resources are important because the data is organized in such a way that you can hook it up to other databases. And the idea is mainly using other people's data, combining it with your data for a new purpose. Right? If you can do that, then you're in business. Right. Sorry. So let's go here. AI, FAIR, fully AI ready is now the acronym, not findable, right? Forget about it. We just do AI. Huh? That's the idea. So, so that's now. I'll get back to some remarks about the future in the few more minutes that I may have, or not even. So I'll flash a few slides with Dutch training initiatives. You see here Health RI, which is the Pan-Dutch health and life sciences center, where a lot is organized. BioSB is the research school, which means we do a lot of training and we do a bit of infrastructure. We try that there. And then there is Elixir Netherlands, which I think I won't explain Elixir. Well, it's a big European project in bioinformatics and with the tagline data for the life sciences. And the Netherlands has also an office there. So Elixir has a training portal test, so to students and others who are interested, there is FAIR data for teaching materials there, which is nice. So BioSB is doing good stuff, but and then there is also for FAIR data, now LIFES in the Netherlands, so there was Go FAIR. The term FAIR has been, Barend Mos was just the guy who was there, he conceived of that acronym the first time. So he has been a tremendous player in all of this. And now he leads this so-called LIFES, maybe Lives, but I misspelled, institution in Leiden in the Netherlands. So the future, quickly. So we're going to scale up even more. The trawler will be even larger in the nets. So superorganismal studies, integrating environment, nutrition and health. Speciality, already alluded to that, combined with modeling techniques. And AI, of course, is omnipresent. See next slide. What is this? First another slide. So here you see all the spatial modeling techniques we had for years. We had POTS models, and so on. So they're all being revisited now and being developed. So you see already various levels of spatial modeling. All of this will now be combined with spatial measurement techniques, and where that will lead, we really don't know, but it will lead to a lot of new insights, probably. So the future of AI. So now it's just use an AI, use a large language model. It knows everything. It's perfect. Already now in the AI community, you see, well, maybe the old Prolog and Lisp programs were not so bad because they could reason in a logical way. And then you see the difference. So LLMs are great, of course, but they lack transparency. They hallucinate, doing crazy things. And systematic reasoning and planning and compositional generalization in general is difficult. So the old-fashioned traditional symbolic AI, as it's called, can do explicit reasoning. They don't hallucinate at all. They can use logic, and explainability is high because it's mainly rule-based. So the idea now is just to combine this. And maybe this is nice that we can shift back a bit from the big trawler, because now we need explicit knowledge as well for reasoning. And maybe the old bioinformatics programs we designed can come into place in combination with all of this, this large-scale stuff. Right. Okay, want to hammer this home. These two medications were fantastic. They were one of the first made by bioinformatics techniques, you could say. Still, the knowledge about the molecules and the proteins was there. So just using AI without further thinking for drugs design is not easy still, I think, to do. So, and that's nice because they will need us as scientists. I'll skip this because you want maybe your coffee. So personalized medicine is one of the important, or a drink, Anton, what is there, sir, in a moment? Drinks. You already? Okay, so, sorry? Yes, that's why I stopped. That's why I stopped. Okay, so personalized medicine, of course, was a dream 10 maybe or 15, 10 years ago, like, oh, the genome, all is in the genome. Now we know, well, what you eat, your lifestyle, the environment, what you inhale is also quite important. And maybe that's even more important for disease onset than all of the genomics. But of course, you know, the combination will be very, very strong. And so you will see developments going on, and hopefully we'll be able to make cancer not a lethal disease, but just a chronic disease, or even solve it. That would be better. Okay, some threats. This morning, and I thought I should show you that UniProt, which was the database that is there to stay forever, is again under threat. And they already started deciding we cannot store all the data anymore, which is a problem. Same is happening for maybe less of a problem. AlphaFold even is now saying we are hitting the limit, not going to store all of our predicted structures anymore. So in the absence of a funding search, which we would need to store all the data and do the science and the combinatorics and all of that. So maybe the idea is, you know, federation, and the FAIR principles support federation of data, doing things locally and combine it on local servers and so on, and have your services like that. But, you know, then we need a few initiatives that support this. And luckily, not in America, but maybe in Europe, you can see that, you know, there will be the European health data space that some in the medical business think it's a threat because it's regulations, but it comes with support. But at least now in 2027, you cannot have a data set anymore in the medical domain that is not fair, or is not connectable to all the resources. There will be law. So that is maybe good for science. And now there is the European Open Science Cloud. Barend Mons always joked about this. The acronym already is wrong because it's not only European, it's global. It's not only about open science. There can be closed science. It's in the FAIR regulation, no problem. And, you know, and it's also not a cloud. It's a lot more than that. So, but anyway, it's an important initiative, I think. And, you know, now they will, and from Holland, we have SERV now as one of the partners, and Elixir as well. So, and more players. There are now 14 or 15 centers or institutions being selected by EOSC, but this will be big with a lot of funding. And, you know, this is really to foster and support federated data and services. So that's FAIR and everything else. And so that will be important. So I didn't do everything, so the wrapping up is not correct anymore. I skipped too much. But anyway, sorry for taking so long, and let's have a few nice drinks. Thanks.